All right, so today we're going to be diving into some pretty fascinating territory, um, physiological psychology.
Yeah.
And you've shared some excerpts with me from the sixth edition of Foundations of Physiological Psychology by Neil R. Carlson.
Right.
And looks like we're really getting into the nitty-gritty of how our brains work.
Absolutely. And what's really cool about this deep dive is we're going beyond just the textbook,
Yeah.
and using your source material as a launching pad to explore some really mind-bending concepts about the brain,
Yeah.
structure, its function,
Yeah.
even how researchers actually study this complex organ.
Yeah, I think everyone's had that moment where they've wondered, like, what's really going on inside my head?
Totally.
And this deep dive should give you some answers. And I think a good place to start would be with the story of Miss S.
Yeah, Miss S's experience is a powerful example of just how intricate and specialized our brains are.
Mm-hmm.
So after having a stroke, she completely lost awareness of her left side.
Wow.
Imagine that for a second.
Yeah.
Suddenly half of your world just vanishes.
Yeah.
This phenomenon, it's called unilateral neglect. And it happens because of damage to one side of the brain.
Oh, okay.
In her case, likely the right.
So it's almost like her brain created a blind spot, but not in her vision,
Right.
but in her consciousness.
Exactly.
What can we learn from cases like Miss S's?
Well, it really challenges this idea that the brain is a single unified entity.
Okay.
Instead, it highlights how different regions are specialized for very specific tasks,
Mm-hmm.
and how damage to one area can lead to such specific and strange deficits.
Right.
Think about split brain surgery, where the connection between the two hemispheres is cut.
Right.
Initially, this was done to treat severe epilepsy,
Yeah.
and while patients seemed mostly fine afterward, further research revealed some really bizarre effects.
Yeah, I remember reading about those split brain experiments, and it's almost like the two halves of the brain were having separate experiences.
That's a great way to put it.
Yeah.
For example, if you show a split brain patient an object in their left visual field,
Mm-hmm.
they might not be able to tell you what it is,
Hmm.
even though their eyes are working perfectly.
Wow.
That's because visual information from the left field goes to the right hemisphere.
Okay.
And without that connection, it can't get to the left side, which is usually dominant for language.
This raises a really interesting question.
Yeah.
If information doesn't reach the language centers, does it even exist for us?
It's a profound concept to consider, right?
Yeah.
It makes you wonder about the nature of self-awareness
Mm-hmm.
and how much of our experience is shaped by our ability to communicate it to ourselves.
Yeah, those are huge questions.
Big questions.
But before we go too far down that rabbit hole, I think it might be good to zoom in on the basic building blocks of the brain,
Okay.
the neurons.
Yeah.
You're probably already familiar with neurons.
Right.
But let's take a closer look at how these tiny cells orchestrate this whole symphony of our thoughts and actions.
So imagine a vast network of tiny trees,
Okay.
each one a neuron sending and receiving messages.
Mm.
They have dendrites, like branches, that receive signals from other neurons. The soma, or cell body, is like the trunk integrating those signals,
Mm-hmm.
and the axon, a long, slender fiber, acts like the roots transmitting signals to other neurons.
So we've got all these neuronal trees forming this intricate forest inside our heads,
Right.
but they're not actually touching,
They're
right?
nice.
There's got to be some way for them to communicate with each other.
Exactly. At the end of the axon, we find the terminal buttons,
Okay.
which form trillions of connections with other neurons.
Wow.
These connections, called synapses,
Right.
where the real magic happens.
Okay.
Neurons don't physically touch. Instead, they communicate across this tiny gap using chemical messengers called neurotransmitters.
It's almost like a whisper being passed from one neuron to the next.
Yeah.
And these whispers can either excite or inhibit the next neuron in line, like a complex chain reaction.
Exactly.
Wow.
It's this interplay of electrical and chemical signals that makes the brain so dynamic and flexible.
Okay, so let's focus on the electrical signal for a second,
Okay.
the action potential, which is this rapid change in electrical charge that races along the axon,
Right.
carrying information from one end of the neuron to the other.
It's like a tiny spark of thought.
So we've got our signal racing down the axon. What happens when it reaches the end at the synapse?
That's where things get even more fascinating.
Okay.
The electrical signal gets converted into a chemical signal.
Okay.
The terminal buttons release neurotransmitters into the synapse,
Mm-hmm.
which then bind to receptors on the receiving neuron,
Okay.
like a key fitting into a lock.
Right.
Different neurotransmitters have different shapes, and they only fit into specific receptors.
Okay.
This ensures that the signal is transmitted to the correct target.
This intricate lock and key system is just amazing.
It is.
So how does the receiving neuron know what to do with the signal?
Right.
Is it always the same response?
Not at all.
Okay.
The receiving neuron integrates all the incoming signals, which can be excitatory or inhibitory.
Right.
Excitatory signals, like those triggered by glutamate, increase the likelihood of the neuron firing its own action potential,
Okay.
while inhibitory signals like those from GABA decrease the likelihood.
Okay.
It's this balance of excitation and inhibition that gives the brain its remarkable ability to process information and make decisions.
And of course, we can't forget about the role of drugs in all of this.
Right.
Many drugs work by influencing the actions of these neurotransmitters.
Exactly. Some drugs, like antidepressants, increase the levels of certain neurotransmitters, while others, like antipsychotics, block their effects.
Right.
Understanding this complex dance between drugs and neurotransmitters is critical for developing safe and effective treatments for various brain-related conditions.
This is all incredibly complex, and it makes you wonder, how do researchers even begin to study these intricate processes?
Yeah.
It's not like we can just peer inside a living brain and watch neurons firing in real time.
Right. It's where the ingenuity of research methods comes in.
Okay.
Scientists have developed incredible techniques to explore the brain, both in terms of its structure and function.
Now, how do they do it? What kind of tools are in their arsenal?
One method is experimental ablation,
Okay.
which involves creating a controlled area of damage or lesion in a specific brain region.
Okay.
By observing the behavioral effects of this damage, researchers can infer what that area of the brain does.
It seems like a very delicate process, almost like brain surgery for research purposes.
It is.
Are there different approaches to creating these lesions?
There are, and each comes with its own advantages and disadvantages.
Okay.
Radiofrequency lesions use a high-frequency current passed through an electrode implanted in the brain. Another technique uses chemicals called excitotoxic lesions, which selectively destroy neurons by overstimulating them.
I imagine precision is key in these procedures.
Oh, absolutely.
How do researchers ensure they're targeting the right spot in such a complex and delicate organ?
It's all about precision.
Yeah.
Researchers use a tool called a stereotaxic apparatus, which holds the animal's head perfectly still and provides a three-dimensional coordinate system for targeting specific brain regions.
So they have a GPS for the brain?
It's like a GPS for the brain.
And different methods for creating these tiny lesions?
Right.
What happens next? How do they actually observe the effects of these lesions?
After creating the lesion, researchers examine the brain tissue under a microscope to confirm the extent of the damage
Okay.
and to see precisely which structures have been affected.
Right.
This helps to link the lesion to the observed behavioral changes.
It sounds like a combination of detective work and incredibly precise microsurgery.
Yeah.
But lesion studies aren't the only tool in the neuroscientific toolbox, are they?
Not at all. Researchers also use a range of non-invasive techniques to study the brain in living subjects.
Okay.
For instance, electroencephalography, or EEG, measures electrical activity from the scalp using electrodes.
Ah, EEG, I've heard of that.
Yeah.
It's used to study brain states like sleep and wakefulness, right?
You got it. EEG is particularly useful for studying those states and for diagnosing seizure disorders.
Yeah.
It's non-invasive, relatively inexpensive, and a widely used technique in both research and clinical settings.
Then there are the imaging techniques.
Right.
I've come across computerized tomography or CT scanning and magnetic resonance imaging or MRI.
Right.
It seems MRI is often considered the gold standard for brain imaging nowadays.
That's right. CT scans use X-rays to create images of the brain,
Yeah.
which are useful for visualizing brain structures and identifying abnormalities like tumors or strokes.
Right.
MRI, on the other hand, uses magnetic fields and radio waves to create much more detailed images.
Right.
It's particularly good for studying soft tissues like the brain and can detect even subtle abnormalities.
And what about positron emission tomography or PET scanning?
Right.
I know it's a bit more complex, but how does it help researchers understand brain function?
PE scans are fascinating.
Yeah.
They involve injecting a radioactive tracer into the bloodstream, which is then taken up by active brain regions. By measuring the distribution of the tracer, researchers can see which areas of the brain are involved in specific tasks.
Wow.
It's a way to see the brain light up as it works.
These imaging techniques are truly remarkable.
Yeah.
But while these methods help us understand the brain as it is now, we also need to consider its evolutionary history, right?
Absolutely.
Yeah. After all, our brains didn't just appear out of thin air.
Understanding how the brain evolved over millions of years gives us valuable insights into its structure and function.
Right.
One key concept in brain evolution is neoteny,
Okay.
which refers to the slowing of development, particularly in the brain,
Okay.
allowing for a longer period of maturation.
So our brains take longer to fully develop compared to other species?
Yes.
What advantage does that give us?
In humans, this extended development period results in larger brains and greater cognitive flexibility.
Interesting.
We essentially stay baby-like for longer, which might sound odd. But it's this prolonged development that allows us to learn and adapt to our environment in ways that wouldn't be possible if our brains matured more quickly.
It's almost like evolution hit the pause button on our brain development, giving us more time to soak up information and develop complex skills.
That's a good way to think about it.
Speaking of evolution, it's interesting to compare our brains to those of our closest living relatives, chimpanzees.
All right.
After all, we share a remarkable amount of genetic material with them.
It's true we share a lot of our DNA with chimps. But even small differences in gene expression can lead to significant differences in brain development and capabilities.
Right.
For example, the human brain is about 3 times larger than a chimp's brain.
Wow.
And even though we share many of the same genes, our brains are wired differently.
Okay.
That wiring is what allows for uniquely human abilities like language, abstract thought, and complex social interactions.
It really highlights how small genetic tweaks can lead to massive changes over evolutionary time.
Absolutely.
And it's those changes that make us who we are.
Right. But it also makes you think, where do we go from here?
That's a great question, and one that leads us into even more fascinating territory.
Okay.
We've laid the groundwork by exploring the building blocks of the brain, the ways it communicates, and how researchers study it.
Right.
Now let's delve into some of the more mind-bending aspects of brain function, like plasticity, the brain's remarkable ability to change and adapt throughout life.
Okay, I'm ready.
Yeah.
Let's talk brain plasticity.
Right.
So we've been talking about how intricate and specialized the brain is, but it's not a static organ, right?
No.
What's this concept of brain plasticity you mentioned?
It's really mind-blowing.
Go ahead.
Brain plasticity refers to the brain's ability to reorganize itself by forming new neural connections throughout life. It's this incredible capacity for change that allows us to learn new skills, adapt to new environments, and even recover from brain injuries.
It's almost like the brain is constantly rewiring itself, optimizing its circuitry based on our experiences.
Yeah, that's pretty incredible when you think about it.
It is.
So it's not just a case of what you're born with is what you get.
Definitely not. Think of it like a dynamic landscape, constantly shifting and reshaping itself in response to the demands placed upon it.
So our brains aren't these fixed, unchanging entities. They're constantly evolving and adapting throughout our lives.
Exactly.
What are the implications of that?
Well, the implications are profound, especially for learning rehabilitation and even aging.
Okay.
It means we have the power to shape and reshape our brains through our experiences and choices.
That's empowering.
It is.
Knowing that our brains are capable of such incredible change gives us a sense of agency over our own cognitive destinies.
Yeah.
Does that mean that by choosing to learn and experience new things, we're actively promoting plasticity in our own brains?
Absolutely. And this brings us back to you and your deep dive.
Okay.
By engaging with this material, learning new concepts, and expanding your understanding of the brain.
Right.
You're actively promoting plasticity in your own brain.
I love that idea.
That's cool. Right.
Every time we do one of these deep dives, we're not just passively absorbing information. We're actively shaping our brains in the process.
You are.
It's like a mental workout.
Exactly. You're strengthening existing connections, forging new pathways, and expanding your cognitive horizons.
Right.
And that's something worth celebrating.
I agree.
Now, taking a step back for a moment, let's consider one of the biggest mysteries surrounding the brain, consciousness.
All right.
What does our growing understanding of the brain tell us about this elusive phenomenon?
That's a huge question, and one that has fascinated philosophers and scientists for centuries.
Well, sure.
We've talked about the different parts of the brain and how they work together.
Right.
But how do we explain the feeling of subjective experience, what it's like to be us?
It's a question that continues to baffle us even as we make strides in understanding the brain. We've identified specific brain regions and processes associated with consciousness, what we call the neural correlates of consciousness. But the ultimate nature of consciousness, the hard problem, as it's often called, remains a profound mystery.
It's almost as if we're trying to use the tools of science to study something that might be beyond the reach of science.
Yet there's a part of consciousness that seems to defy objective measurement.
Right.
While we can observe brain activity and correlate it with certain experiences, explaining how those physical processes give rise to the subjective feeling of awareness is a different challenge altogether.
It's like trying to explain the color red to someone who's been blind their whole life.
That's a great analogy.
Yeah.
It highlights the limitations of our current understanding.
Yeah.
Even with all our advances in neuroscience, we're still grappling with this fundamental question of what it means to be conscious or experience the world from a first-person perspective.
This deep dive has taken us from the building blocks of neurons to the grand mystery of consciousness.
It has.
What stands out to you about the interconnectedness of brain structure, function, and behavior?
What strikes me is the sheer complexity and elegance of the brain.
Yeah.
It's an organ that's capable of so much, from basic sensory processing to abstract thought creativity and even the feeling of self-awareness.
Right.
And yet it all boils down to these tiny electrochemical interactions happening between billions of neurons.
It makes you wonder about the potential that still lies dormant within our brains.
It does.
If we can better understand how the brain works, imagine the possibilities for treating diseases, enhancing learning, and even expanding our consciousness.
The possibilities are truly exciting.
Yeah.
But it's important to proceed with caution and a deep sense of responsibility.
Yeah.
As we delve deeper into the mysteries of the brain, we need to consider the ethical implications of our discoveries and ensure that we use this knowledge wisely.
It's like we're explorers charting a new frontier.
Yeah.
The brain is our final frontier, full of wonder, complexity, and potentially even danger.
For sure.
But that shouldn't stop us from exploring, should it?
Absolutely not. Exploration and discovery are what drive us forward as a species.
Right.
But we must remember to tread carefully, thoughtfully, and with a deep respect for this intricate and powerful organ that is the human brain.
It's a humbling thought, isn't it?
It is.
That we carry around this universe of complexity inside our own skulls.
Yeah.
And we're only just beginning to understand its true potential.
It really is, and what we've explored in this deep dive is just a glimpse into this vast landscape of the brain.
Right.
From the divided brain to the building blocks of neurons, from the electrical sparks of thought to the chemical whispers of neurotransmitters.
Yeah.
We've covered a lot of ground.
We've seen how even small genetic differences can shape the course of evolution, leading to the emergence of the human brain with its unique capabilities. And we've explored the tools that scientists use to probe the mysteries of the brain, gaining a deeper appreciation for the complexity of this remarkable organ.
Absolutely.
Yeah.
But perhaps the most important takeaway is this. The brain is not a static entity.
Right.
It's a dynamic, ever-changing landscape shaped by our experiences and choices.
So we have a remarkable degree of agency over our own cognitive destiny.
They do.
It's empowering to know that we're not stuck with the brain we were born with.
Right.
We can actively shape and reshape it throughout our lives.
Exactly. Every time you learn something new, every time you challenge yourself mentally, you're strengthening connections, forging new pathways, and expanding your cognitive horizons.
It's like a mental workout that keeps your brain fit and agile.
That's a great way to put it.
So as we wrap up this deep dive into the fascinating world of physiological psychology, what's the one thing you hope our listeners take away from this exploration?
I would say the profound interconnectedness of brain structure, function, and behavior. Everything's linked, from the tiniest neuron to the grand sweep of evolution. Each piece contributes to this incredible tapestry of human cognition.
It's a reminder that we're not just passive observers of our own minds. We have the power to shape them, to mold them, and to unlock their full potential.
We do.
So keep asking questions, keep seeking answers, and keep diving deep into the wonders of the brain.
Absolutely.
Who knows what incredible discoveries you might make.
Exactly.
That's it for our deep dive into physiological psychology. We hope this journey has sparked your curiosity and left you with a sense of wonder about this incredible organ that is the human brain.
I hope so too.
Until next time, keep exploring.